Hello, we are Group 8. Today, we will explain VDJ recombination. What is VDJ recombination? To put it simply, VDJ recombination is a type of gene processing. Then, in which cells does it happen? The answer is B cells and T cells. B cells produce antibodies, and T cells have TCRs. We will explain the function of them later. To be more precise, VDJ recombination is the recombination of genes encoding antibodies in B cells and TCRs in T cells. Using antibodies and TCRs, these immune cells respond to a variety of substances. The substances which immune cells recognize are called antigen. The important thing is that one cell can only recognize one antigen. Therefore, to respond to a variety of antigens, it is necessary to have a variety of B cells and T cells. It is VDJ recombination that creates this diversity. Let's start by looking at the growth process of B cells. Antibodies produced by B cells mark bad substances that come from outside the body. Antibodies have specificity and can only recognize certain antigens. Therefore, the number of types of antibodies is huge, and there are about 1 billion types. B cells originate from the bone marrow and mature in the periphery. During this process, recombination of antibody genes occurs, resulting in a huge variety of antibodies. This is VDJ recombination. Next, let's look at the growth process of T cells. Stem cells that come from the bone marrow mature in the thymus, differentiate into T cells. T cells express TCRs on their surface, and when presented with antigens by antigen-presenting cells, they perform a variety of functions, including the increase in cells and cytokine production. We have many types of TCRs in order to respond well to various antigens. The mechanism required for this is VDJ recombination too. Finally, let's take a closer look at VDJ recombination. Gene regions which encode antibodies or TCRs include V, D, and J regions. They are a series of gene fragments, and the genes for antibodies or TCRs are constructed by selecting from and binding together each of these regions. As an example, let's look at antibody. Antibody consists of 2 light chains and 2 heavy chains, and each chain has constant region and variable region. In variable region, there are CDR, which varies among each antigen. VDJ recombination occurs in the gene coding CDR. As for the gene coding CDR of heavy chain, there are about 65 types of V region, 27 types of D region, and 6 types of J region. One of each gene is taken to make the gene, so as many as 10,000 types of heavy chains can be produced. Similarly, about 300 types of variable region of the light chain can be produced. Therefore, even greater types of antibodies can be produced by the combination of light chain and heavy chain. In the same way, a huge variety of TCRs can be produced. This is today's summary. Thank you for watching.